Okay, so in prokaryotes, when the mRNA is made, it can be translated right away. And we'll come to that in the next section. Translation is converting the mRNA into protein. In eukaryotes, though, we have a few other things that have to happen. We call this RNA processing. So the mRNA is going to get chopped up and little bits added to it. So some things are going to happen to it. One of the things we need to talk about are introns and exons. Introns are bits that are going to get removed and the exons will get stuck together. We also have the addition of a 5' cap and a poly A tail to eukaryotic mRNAs. So we start with what we call a pre-mRNA. This contains introns. Okay, what are introns? Well, for reasons, eukaryotic genes have little bits in them that do not actually code for the protein. We call these introns or intervening sequences. These are non-coding portions of the mRNA that need to be removed. Why are they there? That is a very complicated, interesting evolutionary question. As you can see, in this gene, the intron is longer than the rest of the gene. Why make all that mRNA and just cut it out? Well, we don't fully know. That's an interesting question. Some of our hypotheses involve thoughts that introns can help regulate gene expression. They might contain binding sites for transcription factors and things like this. But the short answer is we're not fully sure. We have theories, hypotheses, but we don't really know why they're there. But they are. So we need to get rid of them. The exons are the regions that will actually code for the protein. They have the information to code for the protein. So what needs to happen is we need to remove the introns and stick the exons together. There are also regions at the beginning and end of an mRNA in eukaryotes that we call untranslated regions. Those are just what they sound like. They are not going to be translated into a protein. We will come to those in the translation section, but I want you to be aware that they are there. Okay, so let's start by talking about what gets added to the mRNA. There are two pieces that are going to get added, and they're kind of there to, at least our thoughts are, to help protect the mRNA. So in prokaryotes, mRNAs don't stick around for a long time. They might last maybe 5 seconds. That seems short, but you can make a lot of protein in that time. In eukaryotes, mRNAs are sticking around for hours. And two bits get added to them that help with this stability. The first bit we call a 5' cap. Okay, I'm going to call it the 5' cap. It's technically a 5' methylguanosine cap. It has this special methylated guanosine bit on it. Anyways, it's a 5' cap. We stick it on the front. We think that this helps protect the mRNA from degradation. That's what seems to occur when we have the 5' cap there. Also, on the end of the mRNA, we add a what we call 3' poly A tail. So poly means many. This is just a long chain of A's that are added to the end of the mRNA. Again, why is this there? We think it helps with mRNA stability. This is a long chain, like 200 bases of A's that could be on there. There is some research that suggests the longer the poly A tail, the longer the mRNA sticks around. But that is not universal. So we still don't fully understand the purpose of the poly A tail, which seems silly, but yeah, we don't know sometimes. So, but it gets added. Okay, so those two things get added to the front and the back. We also need to remove the introns, and this seems simple, but it's actually a complicated process. I'm going to show you intron splicing in humans here. It does vary a little bit between the different types of life. Plants do it slightly differently. They have a slightly different recognition sequence, but in the mRNA, we have the exons and then we have the introns. The introns need to be removed. How do you do that and then stick the exons back together? And also, how do you recognize what is an intron? Well, we do see that in splice sites, there are specific sequences, particularly GU at the front and AG at the back. We have a complex called the Spliceosome. It comes in and removes the introns. So the process of splicing is removing those introns and joining the exons together. In this, the spliceosome has both proteins and small non-coding RNAs. These are called small nuclear riboproteins, or SNRPs. That's a fun name for them. they recognize these specific sequences and create this little kind of like lariat structure here. It basically splices out the intron and sticks together the two exons. Okay, cool. Well, here's another potential interesting bit about introns. This splicing process does not always occur the same way. So when we were sequencing the human genome, before we had a sequence of the human genome, we estimated that there would be about 100,000 genes because research had estimated that there were about 100,000 proteins in there. They extracted proteins, spread them all out. There were about 100,000. What we found was only about 30,000 genes in the human genome. So what explains this discrepancy? Well, the identification of this thing called alternative splicing allows us to get multiple proteins out of one gene. So here you can see a gene. DNA, we have the exon regions, and then the intervening sequences, the introns. So we have 1, 2, 3, 4, 5 exons. Alternative splicing allows us to stick these exons together in different ways. Here we see the full thing. Exons 1, 2, 3, 4, and 5 all stuck together. But what if we splice out exon 3? Well, then we get 1, 2, 4, and 5. We get a slightly different protein structure. And how about here, where we don't include exon 4, 1, 2, 3, and 5? Again, a slightly different protein structure. So these different exons encode what we call different protein functional domains. They're little regions that do something. Like here, these regions probably span a membrane. That's why they have all these alpha helices. And maybe these bits are receptors or something like that. So you can see that by sticking the pieces together differently, we get slightly different proteins that might have slightly different functions. So you can get protein A here out of this configuration, protein B here out of this one, and protein C out of that one. So that's how you can have one gene that codes for multiple proteins. That's pretty cool. And evolutionarily, there's some interesting things that happen with splicing. We'll talk more about that later in the course. Okay, so which portion of the pre-mRNA must be removed? Go ahead and pause the video, think about that. Okay, the bit that needs to be removed is the introns, those intervening sequences. We remove the introns and stick the exons together. So eukaryotes need to process their mRNA in there. One of the big things is splicing. So we're removing the introns, the intervening sequences, they get spliced out. Then we have the exons, the coding sequences get stuck together. We add a 5' cap to that and a poly A tail. The spliceosome is this mixture of RNA and protein that removes those introns. And we see alternative splicing uses different combinations of exons to create slightly different proteins. All right, that's it for our transcription and mRNA bit. In the next section, we'll talk about protein synthesis.